Members of the Academy, distinguished guests, ladies and gentlemen, allow me to speak about science rather than the human heart. It's an enormous honor to be selected as one of this year's Nobel laureates in chemistry. I would like to thank the Royal Swedish Academy of Sciences for bestowing the Nobel Prize on me, along with my co-laureates Richard Henderson and Jacques Dubochet. The thought of being considered a peer of Ernest Rutherford, Marie Curie, and Linus Pauling is truly daunting. I developed an interest in electron microscopy when I worked with Ernst Kinder on I developed an interest in electron microscopy when I worked with Ernst Kinder on my master thesis in physics at the University of Munich. My thesis had something to do with backscattering of electrons. In 1943, working with the electron microscope, he had studied butterfly wings, which, as he realized, gained their brilliant colors from interference of light on gratings formed by tiny scales arranged in regular order. I signed on to a graduate project with Walter Hoppe at the Max Planck Institute in Munich, an X-ray crystallographer who had developed an interest in electron microscopy as a means to study biomolecules. He viewed the electron microscope as just a diffractometer that, unlike the one employed in X-ray crystallography, could record not just amplitudes of diffracted electrons, but their phases as well. This was just a fancy way of saying electron microscopes were able to form images. For the beginning years from 1930s to the 1950s, the contributions of electron microscopy to biology were confined mainly to the investigation of tissue. Serious forays into the quantitative visualization of molecular structure did not start until the 1960s and were concentrated in three groups: Aaron Klug at the Lab of Molecular Biology at the MRC in Cambridge, my mentor Walter Hoppe at the Max Planck Institute in Munich, and Edward Kellenberger, as you heard, at the Biozentrum in Basel. Now, unless symmetries are present, three-dimensional reconstruction of an object requires the combination of its projections from a wide angular range. First pioneering achievements in molecular structure research with the electron microscope were the three-dimensional reconstruction of the bacteriophage tail with helical symmetry in 1968 by de Roussier and Krug, and the first reconstruction of an icosahedral virus in 1970 by Tony Crowther in the same lab. At that time, biological molecules could not be imaged in a close to native state. Negative staining, which amounts to embedding the molecule in a layer of heavy metal salt, was the only means available to produce contrast. On the other hand, biological molecules are quite fragile, and it was known that maintenance of their integrity would require a fully hydrated environment. When I started my work as a graduate student under Walter Hoppe, I was exposed to discussions in small workshops in the Tyrol Alps, co-organized by Walter Hoppe and Max Perutz in Alpbach, and once in Hirschegg. These were the first meetings that brought together protein crystallographers and people working in electron microscopy. The many problems faced in attempting to image biological molecules in the electron microscope were discussed at a workshop organized by Edward Kellenberger and Geis in the Swiss Alps in 1973. The state of the art at the time was reflected in the title of a proceedings paper, since high resolution was equated with any results better than 30 angstroms. Paramount at the meeting was the search for a method that would keep the molecule fully hydrated while exposed to the electron beam. following pioneering studies by Bob Glaser in 1971. Just at the time I visited his lab as a Harkness Fellow, radiation damage was recognized as a major obstacle in the strive toward higher resolution. Averaging over a large number of repeats of a structure exposed to very low dose was seen as a general solution to this problem. Thus, this meeting set the stage for a groundbreaking study by Richard Henderson and Nigel Unwin that you have already heard about in 1975, the reconstruction of bacteriodopsin from the purple membrane of Halobacter embedded in glucose under quasi-native conditions. The confluence of novel approaches to sample preparation, data collection at extremely low electron dose, and merging a tilt series of these noisy images made this work a towering achievement. The time after that was marked by general excitement in the community and many attempts to use the same or similar methods in the study of other proteins amenable to two-dimensional crystallization. However, the intrinsically low contrast between proteins and glucose made these attempts difficult, and embedment in ice remained the agreed general goal. At the time, most researchers in the field that was to be called structural biology were united in the belief that serious structure determination required highly ordered samples, such as 2D crystals, helical fibers, or viruses with high symmetry. Attempts to extract structural information from freestanding single asymmetric molecules were not taken seriously. One such alternative approach pursued by Walter Hoppe was to tilt the EM grid to which isolated molecules were attached in the multiple angles while collecting projections from which the molecules could be reconstructed. While the angles are exactly known in Hoppe's tomographic approach, the accumulation of radiation damage over multiple exposures to more than 1,000 electrons per angstrom square at the time rendered the end result essentially meaningless. The other approach was the one I formulated in a concept paper in 1975, which I will elaborate in the following. So this approach takes advantage of the fact that biological molecules purified from cell extracts and suspended in solution typically exist in thousands or even millions of copies, that is, separate realizations of the same structure. Hence, if a grid to which such a sample has been applied is put into the electron microscope, a large number of projection images of the same molecules lying in random orientations are encountered. The important point is that instead of collecting multiple images from one molecule by tilting it in the electron microscope, one merely has to take a snapshot of multiple copies of that molecule with a very small radiation dose. The advantage in the latter case is, of course, that the 3D image obtained relates to a molecule that has seen only one single low exposure and has remained practically undamaged. The terms single particle averaging and single particle reconstruction, which were later coined, refer to the fact that the molecules in the sample are freestanding and not attached to one another as in a crystal. So at this point, it is important to reiterate that no symmetries are assumed in this approach. Rather, the idea was that entirely asymmetric molecules could be reconstructed in this way. Symmetries obviously simplify the problem, as in that case, the image contains multiple projections of the repeating unit in different known orientations, allowing data from different molecules to be readily merged. Now, simple as the concept sounds, the realization of single particle reconstruction required several problems to be solved, all in the area of image processing. First, is it feasible at all to align noisy molecule images with one another with sufficient accuracy? Second, how do we estimate the resolution of a reconstruction obtained by combining all images? Third, how do we sort molecule images by appearance related to view angle and conformation? Fourth, how do we find the viewing angles of projections a posteriori? Fifth, how do we reconstruct molecules, a molecule from projections at randomly spaced view angles? So it was in the nature of these problems and their novelty that progress was slow and piecewise, one step at a time. During the work on my PhD thesis, which was 1968 through 1970, I had already explored the use of cross-correlation function for aligning electromicrographs. I discovered that two successive images of the same area of carbon could be aligned with a precision of better than 3 angstroms. Actually, the process of 2D alignment is a bit more complicated since molecules picked from the micrograph differ both in shift and rotation. The solution I found to determine both relative rotation angles and shift simultaneously makes use of the fact that the autocorrelation function of a molecule is shift invariant. It does not depend on its position within the image frame. Then working with Owen Sexton at the old Cavendish lab in the group headed by Vernon Coslet, I was able to show that alignment via cross-correlation would be accurate enough for the purpose of aligning noisy images of single molecules if the dose exceeded a certain level, which depended on the molecule size and contrast against the background. Using the ribosome as an example, it became clear from the formula we obtained that single-particle approach to structural research was indeed feasible for molecules of sufficient size. My appointment in 1975 as senior research scientist at the Division of Labs and Research, later named Wadsworth Center in Albany, offered me the opportunity to explore the idea. With the help of samples provided by collaborators, I was able to demonstrate the feasibility of obtaining two-dimensional averages showing enhanced features of molecules with images of glutamine synthetase, acetylcholine receptor, and 40S ribosomal subunits from HeLa cells. Among these, the 40S subunit averages were most striking in showing the potential of the single particle technique and proved instrumental for gaining funding by the National Institute of Health. Nonetheless, presentations of single particle averaging results at the meeting at Burg-Giemen in 1979 by myself and my collaborators Martin Kessel and Peter Singsheim were greeted with a great deal of skepticism. One issue to be addressed, which I had already mentioned before, was the fact that due to the absence of crystal order, the average of aligned molecule images shows no diffraction spots in its Fourier transform, and therefore lacks an inherent measure of resolution. Without such measure, progress in quality cannot be tracked and compared among different groups. Following an earlier study during my dissertation on the effects of drift on electron micrograph, I realized that signal bandwidth is reflected by the extent of reproducible information in Fourier space. This extent of reproducible information is apparent from Young's fringes that show up in the optical diffraction pattern when two successive micrographs of the same specimen field are superposed with a slight shift applied. How can this idea be translated into computational procedure? Reproducibility can be quantified by dividing the data randomly in half, then comparing the Fourier transforms of half averages over rings in Fourier space. Resolution is then defined as the ring radius where a measure of comparison, for instance, the phase residual or R factor or cross-correlation, passes a critical threshold. The same measures computed over shells would later prove important in estimated resolution of 3D reconstructions as well. These first studies of image averaging immediately brought up the problem of heterogeneity. Only those molecule images could be combined in an average if they originated from molecules of identical structure and showed the same view. At that time, one of Ernst von Bruggen's students, Marine van Heel, visited my lab with images of Limulus polyphemus hemocyanin, which is an oligomer with a distinct architecture producing multiple preferred views when negatively stained and imaged in the electron microscope. These images therefore presented a perfect example of heterogeneity. The solution to this problem came from the insight that images, once aligned with one another, may be regarded as vectors in a space of n dimensions, where n is the number of pixels. Groups of images that are similar will show up as clusters of vectors in that high-dimensional space. Equivalent problems of finding clusters in high-dimensional space were encountered in many fields of science and gave rise to multivariate statistical analysis, a procedure which determines a compact, low-dimensional subspace tailored to the problem. With the help of Jean-Pierre Breteaudier, a Wadsworth Center scientist working in laboratory medicine, we were able to use a program meant to sort blood samples to sort images instead. Application to hemocyan proved a great success. Early on, as I set out on this new approach to recovering structure, it became also clear to me that in order to make systematic progress in the development of algorithms and computer programs with ever-changing and expanding goals required a workbench with a large set of tools. To this end, I developed a modular image processing system called Spider, which made it possible to design complex programs from pre-coded building blocks using a simple script language. For example, the command WI would invoke a routine for extracting a rectangular portion of an image, and AC would compute the autocorrelation of an image. All programs were coded in Fortran, the most advanced language at the time. Most of the initial programming was done by a student of computer science and one undergraduate. The script language of Spider then became literally the lingua franca in my lab, and as the suite was disseminated to other labs within a growing community of users. I attribute the idea underlying the Spider system and its modular design to my stay at a jet propulsion laboratory in 1970 under the Harkness Fellowship. During this one-year visit, I learned to write program modules that interfaced with Vicar, the image processing system in use by NASA since 1966 for analyzing images from planetary missions, such as the unmanned Jupiter flyby mission. Now to the subject of three-dimensional reconstruction. For computing the three-dimensional structure of an object from its projections, one requires the angles of each projection to be known. Determination of the angles of the randomly oriented molecules was the most important yet most difficult problem to be solved. The solution came from the insight that two micrographs, one of a field of untilted particles, one of the same field tilted, contained all the information required to assign Eulerian angles to each tilted particle. In this geometry, the angles of the tilted particle projections lie on a cone with random azimuths, giving rise later to the term random conical reconstruction. In 1982, I was joined by Michael Rademacher, also a student of Walter Hoppe, who had worked in his dissertation on algorithms for 3D reconstruction from projections arranged in regular conical geometry. Thus, he had exactly the skills needed to develop computer programs that implemented the random conical reconstruction. One important step was still missing: the generalization of the algorithm that assumed regularly spaced conical tilting to the general case of random angles. Once this was accomplished by Rademacher in 1986, we obtained the first single particle reconstruction using the random conical method, the 50S subunit of the E. coli ribosome. This reconstruction was limited in quality by two factors. One was the missing cone of information in the three-dimensional Fourier transform, a source of unidirectional artifacts in the three-dimensional density map, and the other were the artifacts due to the preparation of the sample by air drying with negative stain. Both were readily overcome at the time within a short period. The missing cone problem was solved by merging data sets obtained with 3 or more different zero-degree views, and the preparation of the sample with negative staining was replaced by cryo-embedding in vitreous ice, following the spectacular success of Jacques Dubochet's vitrification method in the application to viruses in 1984. Yet another important problem affecting the quality of all reconstructions from EM data is the distortion of information by contrast transfer function, or CTF, caused by the lens aberrations of the electron microscope. This problem and its effect on the reconstructed density maps was eventually overcome by my group and by others through merging data obtained with different defocus settings using a Wiener filter algorithm. The first molecules visualized by a cryo-EM and reconstructed in 3 dimensions with a single particle method were the E. coli ribosome, hemocyanin, and calcium release channel. A cryo-EM reconstruction of the haloacular Maris mortuae ribosome proved to be helpful in the phasing of the first X-ray structure of the large subunit. All final reconstructions benefit from an iterative angular refinement that starts out with the first rough reconstruction. In any project dealing with a molecule whose structure is unknown, it is practical to distinguish between 2 phases, a bootstrap phase and a refinement phase. In the bootstrap phase, a first rough reconstruction is obtained either by the random conical method or by an alternative method developed by Marie van Heel and others, in which common lines in Fourier space are employed. In the refinement phase, an existing reconstruction is used to generate a library of even space projections with which the experimental projections are compared to assign refined angles to them for the next round of reconstruction. Now I would like to show a few examples for how the new technology, well before it's reached the present state of perfection, gave us important insights into pivotal processes of translation by the ribosome. In the late 90s, one of my postdocs, Rajendra Agrawal, prepared a sample containing elongation factor G bound to the ribosome as it catalyzes translocation of mRNA and tRNAs. Comparison of the cryo-EM reconstruction with that of the unbound ribosome showed a dramatic change. The small subunit had rotated by almost 10 degrees. This finding provided first clues on the mechanism of mRNA-tRNA translocation in this important molecular machine. In 2002, another postdoc, Michael Walle, In 2002, another postdoc, Michael Walle, found that in the decoding process, tRNA entered the ribosome in complex with protein factor EFTU in a strongly distorted form, in the so-called AT state. This observation indicated that the tRNA acts as a molecular spring, apparently setting the threshold for discrimination between cognate and near-cognate codon-anticodon pairing. As a third example, in 2001, we teamed up with Jennifer Doudna, then at Yale, to visualize the mRNA from hepatitis C virus in the process of hijacking the human ribosome. My postdoc, Christian Spahn, working alongside with Jeff Kieft from the Doudna team, visualized the so-called iris element of the virus attacking the small subunit of the ribosome. Even though before 2013, when the cameras came out, cryo-EM reconstruction fell short of resolutions in the 3D angstrom range. It was still possible to interpret them on the basis of existing structures by means of flexible fittings, such as MDFF, deriving interpretations through atomic models. I'm showing two examples among many, a 6-point angstrom cryo-EM map presenting a snapshot of the decoding process and its interpretation by Elisabeth Villa, and the atomic model of the ribosome from T. Brucey, built from a 5.5 angstrom map by Yasser Hashem, still from data recorded on film. I finally come to a distinct advantage of single-particle cryo-EM that is brought out in many applications now. Here, unlike in X-ray crystallography, molecules are unconstrained by crystal packing and are able to assume the full range of conformations present in solution. For a processive molecular machine like the ribosome, unless it is stopped by a chemical intervention, many different states coexist in solution. The full potential of single-particle cryo-EM has been realized with the introduction of maximum likelihood classification by Sjors Scheers, who worked as a student in the lab of Jose Maria Carrazzo and later independently at the LMB MRC in Cambridge. These algorithms perform the seemingly impossible task of disentangling changes of view angles from changes in structure. A large, well-characterized ribosome dataset that we supplied was correctly classified in the initial study. Working with this toolset, multiple structures are now routinely recovered from a single sample. In the next slide, shown, I show two examples. Several states of the translation elongation cycle are fished out from images of ribosome purified from a cell extract of Plasmodium falciparum, the malaria parasite. The second example on the right is another study by my group where part of the elongation cycle is visualized in an example containing a mutant of elongation factor G. Literally, cryo-EM is now able to tell us a story from a single sample, showing how the molecule changes its shape and binds smaller molecules as it performs its work. Ultimately, even the recovery of a continuum of structures reflecting the states of a molecule at work and the mapping of the free energy landscape are no longer a distant goal. Even though I have illustrated the progress achieved by using my favorite molecule, the ribosome, which now has the best resolution of 2.5 angstrom, the range of applications in biology is virtually unlimited, except for some lower bound on molecule size, and of course, the necessity of having the molecule suspended in solution. My own recent collaborations with the groups of Andrew Marks, Wayne Hendrickson, Alexander Sobolewski, and Filippo Mancia demonstrate the gain in knowledge achievable now for membrane-bound channels and receptors. To conclude this account of a 40-year journey, I must confess that even though I was always a firm believer in the technique I dreamt up many years ago, I never thought it would see it and come to fruition in this spectacular way, let alone that it would earn me a share of the highest prize coveted by scientists all over the world. It has been a truly fantastic journey overall. The knowledge that human medicine as a whole stands to benefit from the new technology is ultimately the best reward. I should at this point give tribute to all the people who have made this development possible over the years. It goes without saying that without the pivotal contributions by my co-laureates Jacques Dubochet and Richard Henderson, the field would not have developed and prospered in this way. My thanks go to many students, postdocs, and collaborators who shared my vision and put in so much hard work to make it all happen. Bob Grasucci has been with my group for more than 30 years in management and support of instruments with ever-increasing complexity. My very special thanks goes to my wife, Carol Saginaw, who has steadily supported me for close to 40 years and never lost faith in me, and my family and friends who have cheered me on at every turn. Much of the early developments of the technique took place at the Wadsworth Center in Albany, which provided a sheltered, supportive environment with generous funding by the state of New York, particularly in the starting phase. In my expression of gratitude to the Wadsworth Center, I would like to single out Carmen Mandela, who supported me throughout those years as a colleague, collaborator, and friend. My affiliation with the Department of Biomedical Science at the University at Albany since '85 allowed me to foster academic interactions with SUNY faculty at large. And since 2008, when I joined the faculty of Columbia Department of Biomedical Sciences and Biological Sciences, I enjoyed multifaceted intellectual environment fostered and maintained by one of the country's greatest universities. My work has been generously supported by the National Institute of General Medical Sciences of NIH, almost without interruption since 1981. Funding for 2 inspiring sabbatical stays in England and Germany was provided respectively by the Fogarty Foundation and the Humboldt Foundation. I received sizable awards from the National Science Foundation for several instrument purchases, and support by the Howard Hughes Medical Institute for almost 20 years has allowed me to build up a state-of-the-art, professionally supported cryo-EM facility, first at the Wadsworth Center and later at Columbia University. Thank you.
Could I please ask our co-laureates thank you very much.